Okay, welcome back. Thank you very much for staying in this with us. It's been a fascinating workshop thus far, and we look forward to continuing it. It's a real pleasure for me to introduce Dr. Elizabeth Thiele, who's a neurologist and epileptologist at Massachusetts General Hospital, where she is the director of the Pediatric Epilepsy Program and a professor of neurology at Harvard Medical School. Dr. Thiele's research and clinical interests include the role of diet in epilepsy treatment. Dr. Thiele.
Thank you very much, Adam. And I'd like to thank the organizers for including me in this. I think this is a fascinating meeting with the pre-recorded talks and then these discussions. So you'll see our challenge here is to discuss clinical research and what are the challenges to trial design and their solutions. And to quickly introduce the panelist, Jackie French is professor of neurology in Comprehensive Epilepsy Center at NYU Langone School of Medicine, also founder and director of the Epilepsy Consortium. Jordan Elm is an associate professor of biostatistics in the Department of Public Health Services, Medical University of South Carolina. Helen Cross is the Prince of Wales Chair of Childhood Epilepsy and Health and head of the Developmental Neuroscience Research and Teaching Department at Great Ormond Street in London. Bobbie Barron is a research dietitian at Johns Hopkins University. And then Glenna Steele, importantly, is a founding board member and the first president, as well as the first education director of the GLUT1 Deficiency Foundation. So the pre-talks that were recorded, I think, looked at the concept of trials in dietary therapy through different perspectives. Helen gave a beautiful overview in her talk about the clinical trial experience in dietary therapy. I'm talking about the trials that have been done and their limitations, and also the inherited biases in many of them. And she broke down the concept of trials into different categories that I really liked, such as intervention, what would be the trial that was being performed, what would be the treatment, who would be the comparator group. Blinding, we know, is a very difficult and complicated issue in dietary therapy, so what aspects of trial could be blinded. Recruitment, in many of the trials, recruitment has been slow and difficult, and also compliance. How do we judge compliance with the dietary treatment? Also, how do we retain patients in the trials to make sure we get adequate power for results? What are our outcome measures, be it efficacy with regard to seizure or other issues? What is the optimal duration of the trial? And what type of follow-up should be arranged? And then Jackie's talk, being the guru of epilepsy clinical trials, I think can help us take a step back from the trials that have been done with more the conventional randomized controlled trials or open label trials. And she talked about master protocol trials, the new concepts of the basket trial, umbrella trial, and platform. And would these be appropriate models to use in pursuing dietary trials? Jordan then importantly also looked at the importance of statistics in powering our trials and the importance of having a pre-specified statistical analysis plan. We know that the issues in metabolism-based therapies with regard to trials are tough, again, because of the blinding, the issue of compliance, and also how do we design a trial to have sample size calculations that would adjust for non-compliance, and how do we handle the concept of missing data. Bobbie gave a very important view from a research dietitian perspective of what are the types of food or nutritional trials that can be done. Many of the trials that have been done have been diet counseling, providing the family with the education or starting the diet and then having them follow it, versus the concept of feeding styles. Could you enhance compliance by actually giving the participants in the trial their foods for the duration of the diet? She also discussed how do we assess compliance in a diet-based trial where we can't measure drug levels or other things, and the concept of following closed food records that the dietitians would analyze to really determine was the patient adhering, what was the diet the patient was receiving, and also the value of following ketone levels. Whereas for us with low glycemic index treatment, that would not be as helpful because we often do not get an elevated ketone level. And then I think very important was Glenna's talk from the patient perspective and the patient advocacy perspective. And I really liked her phrase. It says, nothing about me without me. And I think a lot of the epilepsy trials are now stepping back and saying, look, we're not just trying to treat seizures in these different disorders, but improve overall outcomes, level of functioning, behavior, cognition. And I think working closely with the patient advocacy groups is going to be really key as we move forward, seeing what their needs are, what their goals are, and helping all of us achieve the common goal of improving epilepsy in these different patient populations and overall quality of life. So with that and kind of the overview of those talks, I think what we'd like to discuss today, just to kind of thoughts before we have the panelists speak, was next slide please, is what questions can be and need to be answered by clinical trials. Another is, are there non-traditional clinical trial designs, like Jackie mentioned or others, that might be better suited to metabolism-based therapies in studies in epilepsy? And then how can principles of rigor be adapted to metabolism-based studies, given the inherent limitations, again, the lack of the difficulty blinding, et cetera? And what would families find acceptable to participate in these trials? Would a feeding trial work in these patient populations? Then, and I know this will be a discussion in the next panel as well, but I thought it'd be good to give these panelists an opportunity to comment on this, is how can clinical trials inform the basic science studies? What can we learn from the clinical trials we're doing and what we're seeing in the care of these patients that help advance basic science? And then finally, and I think very importantly, how can we partner and work with patient advocacy groups, including choosing outcome measures that are important to these populations in addition to improving seizure control, and also to help optimize recruitment. Glenna made a very important point in her talk. These patient advocacy groups are often very strong communities of very committed patients and families. And if we look at the recent pharmaceutical trials in pediatric epilepsy, both in Dravet syndrome and Lennox-Gastaut syndrome and in tuberous sclerosis, I think it's the patient advocacy groups that really helped those trials enroll quickly and generate a lot of interest in those trials, as well as education about the possible therapeutic benefits of the therapies. So kind of with that, what I'd like to do is for the panelists, maybe have you address any of these questions and maybe give one recommendation or suggestion as how you think we could move the field forward by making future clinical trials more rigorous and unbiased. And Helen, if okay, I'll start with you. You have a lot of experience both in using and studying the ketogenic diet in the treatment of refractory pediatric epilepsy. So what is your view on the future of clinical trials in dietary therapy?
Thank you, Elizabeth, and thank you for asking me to participate in this. And I acknowledge that I think my talk dealt with the problems that we've had and probably provided more questions than necessarily answers. This aside, I'm thinking of addressing some of your questions. I've spent many years, as you have, trying to justify that we do need more clinical trials. Often there is a question as to, well, surely we know it works. And I think probably we would acknowledge that for a full ketogenic diet in childhood epilepsy, we do have a lot of data to suggest that it works. We have, several randomized controlled trials, and the difficulties that have been expressed with regards to where we perhaps still need answers is in adult trials. There are some trials, but recruitment has been really difficult. And this is where an area where perhaps there does need to be a degree of evidence based because of the need for resource in order to provide for that. But maybe it's not just efficacy, but how we enhance compliance and well, not necessarily compliance, retention, and make it more acceptable. And does the trial or the primary outcome in an adult trial necessarily need to be efficacy? And maybe that could be raised. Interestingly, of course, when it comes to trying to recruit to a trial, my experience has not been that necessarily the major problem is that families don't want to commit to a dietary therapy, but it's actually the fact that they've come to the stage that they feel dietary therapy is the only way forward, and therefore they don't want to be randomized. And this is particularly my experience in the very young population. So it's designing trials that perhaps can recruit earlier in the process. And this is where discussion for design, I think, with families and patients really comes to the issue. But the two other issues I just wanted to raise, well, three actually, I wanted to raise was one, should we be looking at the full dietary therapy in the light? I mean, although I may remain a little even more confused with the biochemistry and everything that we've heard about within this workshop. I think it's a very different approach if we're going to try and test compounds. And is it just one compound, or is it an additional issue, so that we want to try different combinations as opposed to a full dietary therapy. And I think that there it does become the issue with the full dietary therapy, how you blind administration, how you blind outcome, and indeed how you ensure consistency. But the other issue is, of course, we've heard that there are multiple mechanisms of action. Is it the same mechanism of action in each type of etiology or epilepsy? And many of the groups where we've looked at ketogenic diet therapies, they're non-homogeneous groups of patients. And therefore, perhaps we have to, in order to look at more rigor, we need to look at more homogeneous groups of patients. Perhaps that would mean that we don't need such a large sample size. That would need to be looked at. But just to look at consistent groups so that we're not looking at multiple mechanisms. And perhaps we have to look at what we've done before in order to look at the groups that we should be looking at moving forward. And then it comes to the whole issue about outcome. And we've always looked at seizures. Of course, we've looked at seizures, but one way forward with some of the more complex groups is looking at what outcomes matter to the families, or the patients and families. What outcomes? How can we look at perhaps even a composite outcome? Is it only seizures? And this is where we come to: can we blind outcome assessment? Because even if we have seizure diaries, then we're still relying on counting, and there'll still be a degree of bias as that families aren't necessarily biased to the treatment. So even looking at a blinded outcome assessment, we're still looking at seizure diaries that have not been blinded in the way they've been recorded. So then we need to think of, is there something else from an outcome point of view, maybe a biomarker, but we've just discussed that we're not there yet. In order to try and add to the outcome frame as the outcome measures that we could blind for it, whether it be degree of attention. And if we had homogeneous groups of patients, then maybe there would be a outcome measure that could be consistent to that group that could be added to the seizure outcome.
No, thank you. And I definitely agree. I think if you look at the pharmaceutical development recently, it's really been targeted more to specific disorders. With Dravet, with TSC, granted not one disorder with LGS, but kind of more specific, trying to address areas where there's significant unmet needs and see if there could be, some specific signal of efficacy. I mean, we were very, very impressed when we started using low glycemic in Angelman kids to see how incredibly effective it is in that population. So Jackie, then turning to you kind of as the guru, I consider the big guru in epilepsy clinical design and trial experience. I really like the new protocol designs. And do you think that either the platform or the basket trial designs could help us with dietary therapy? Like the platform to say, we have several different diets. People have wondered, should we compare them? Should there be a comparison between low glycemic modified Atkins, traditional keto, and would the platform design allow us to do that if we thought that was important? And also the basket trial, like you said in your talk, like Takeda Abed now doing a basket trial to where we could see, is there particular signal in some of these rare epilepsies where there really are not effective treatments?
Yeah, I mean, what's kind of interesting is you could almost use a basket trial as a validity measure, because if you did find that there were some segments of the population that were super responsive and others that were not, and obviously the population wouldn't know that, all of them would be equally as likely to be biased. If you see a huge signal based on the type of epilepsy that you're looking at, that might at least give you an indication that, not only was it working in that group, but also that it was working better. And unless you could find some reason to imagine a bias in that way, that might be very informative. We do a lot of basket trials now, and I think Helen was kind of indicating that when she was saying we need to look for more homogeneous populations. But I think that, rather than maybe looking for only homogeneous populations, we have to subdivide, as Elizabeth was saying, so that we follow a group and we have that, as Jordan would say, pre-specified into the outcome of how we're going to handle each of those groups. So I think that would be definitely very interesting. I heard a great talk by Gary Gronseth at the American Neurologic Association, where he was talking about quote unquote real world data, and whether, you know, there was some way to interpret real world data. And basically we always come back to the issue of the parachute, right? So, you know, his point was that if you have some, you know, situation where the outcome has been the same 100 out of 100 times, and you know, you can then go in and make sure that you are creating that same situation again, you know, that, you know, there are scientific ways to say if you get a completely different outcome after you do A, B, or C on the 101 to the 115th time, then that's interpretable. So, I mean, I think that we focus a lot on randomization and blinding, and all of those things are very important, but they also can give you skewed answers, and they also can give you biased answers. And I think that as a population, as a scientific population, as scientists, we have to really focus on the science of interpreting non-blinded data. And maybe we haven't put all of our, you know, thinking caps on in regards to doing that. And I would love to hear what Jordan has to say about that.
Yeah, Jordan, jump on in, especially kind of looking at it as a statistician and being familiar with the diets that have been done and the potential sources of bias and the challenges we have with keeping these blinded. And how do we measure compliance? So what recommendations would you have for us in making our future diet trials more rigorous and unbalanced and successful?
I think in terms of blinding, that's a very hard one. I mean, I think it really comes back to what Helen was saying about finding some way to measure in which you have a blinded outcome assessment done. And I don't really know the specifics of how that would be done in this context, but it seems like that could definitely be something that is developed. If that can't be done, then finding a more objective outcome. I don't know if that has to do with, you know, EEG measurements of seizures or something like that you could assess in the home. You know, maybe the patient wears a device at home, and then that's a more objective assessment. You know, that would be something that would deal with the blinding. But that's, I think that is, you know, I think that's something that the field needs to spend a lot of time to really think about, because that's going to be important. The other part of it deals with the process of randomization itself, and I think who gets selected into the group, and is it at least the allocation blinded, so that you're not picking the people who are the most likely to maybe comply with the ketogenic diet into that group, you know, so that the process in which patients are getting randomized is being done, at least that part's being done blinded. I think in terms of the study design, I love the idea of the platform design as well, with potentially having multiple active treatment arms in a study. It sounded to me, just from listening to some of the other talks, that there was quite a few different types of interventions, and including not just dietary interventions, but exogenous ketones, whey, decanoic acid. And some of these things maybe are at the stage where they could be combined into a large platform trial. Now, some of them may not be there yet, and they might need some more early phase studies, tolerability studies. And so it kind of, I think it depends on how many are at the right point, but that would lead to a really interesting question, I think, which would be about how would you define your control group if you have multiple interventions? And I think it would be nice to have a control group in which you had, you know, a smaller, you know, so you're not having, you know, a control group that's equal to the same size of each active arm. So it's a much smaller control group. I think that would make it, you know, enticing to patients to participate in such a trial, where you have the more of a higher probability of getting into an active treatment arm. But then your control group might be a little different for different people. So if you, some part of the control group might be a control for the diet, and then you would pool that with people who are also maybe in a control for a placebo for some other type of pill or whatever it is that is an injection or something like that. But so I think there's a lot of fun ways that you could think about designing such a platform.
Thank you. And then Bobbie, this is what you do for a living, kind of the whole concept of research and dietary therapies. And so one of the talks mentioned that even in a control group, and we see this in clinical practice, if we talk to families about starting a diet, sometimes they'll start implementing changes in what the child eats even before we officially start a diet. And that's a source of bias and also could confound the results. So what do you think, kind of moving forward with your experiences on where you've been, what you've done, how do we really design trials with dietary therapy? I think if it's a trial of something like whey or other things like that, that might be easier to have, a placebo, et cetera, and it might be easier to blind. But what would you recommend to us as a community to move this forward with future trials?
Right. I think after hearing all the talks today, we have an appreciation for how complex the metabolism can be. And when you're dealing with a supplement like whey protein, or you're dealing with MCT oil, even that alone can have a cascade of effects. So when you're dealing with a diet, it's even more complex. And so I like the idea of the platform design where you can look at multiple diets and also have a control diet. So in my talk, I talked about diet counseling studies and feeding studies, and diet counseling studies is what we use in the clinical practice and in research too. And we know that there are very strict, the classic ketogenic diet is very strict, but we also have the more liberal versions. And in classic keto, the food variety is very limited and the day-to-day variation is very limited. And so the education is almost what you get. Yes, there are going to be lots of differences, but what you are educating on is close to what the patient's eating. Whereas we have the more liberal versions of the diet, and the education can be one thing, but because compliance is such an issue, we take lots of liberty and freedom to adapt the diet to that individual. To make it work for them, to make it sustainable for them. So what we're actually educating on in the beginning, let's say a 20-gram net carb diet, could be very different from what they're actually eating. And that's not necessarily a bad thing in clinical care, but when we start talking about research and standardizing these diets, it really muddies it up. So I also talked about using feeding studies as a way to control that. And they're very labor intensive. They take a lot of time to plan on the dietitian side, but they are doable and they're typically feasible. So incorporating that into a platform design might take advantage of, the different diets, the time it takes to plan them. So in regards to blinding, we've learned from previous studies that there are lots of challenges regarding it. We learned from the Hopkins study that fasting potentially played a role in the outcomes, and we know now we don't need to fast, so we can exclude that. We have lots of foods from formula companies that are ketogenic and that can mimic foods. Dietitians, they study food and we can take a lot of liberties and be very creative with how we could blind actual feeding studies. There are, ketogenic formulas that can be blinded to. So I think we do have some options, and I think it's worth exploring blinding. But again, in feeding studies, they're usually very short term. So we would have to think about what clinical questions what questions we want to answer with potentially 3-4 month trial. Oh, and sorry about the control diets too. We have a variety of diets that we could use. Typically it's like their standard diet, so we would assess that by food records. But if we want to actually administer a diet, control a common control diet in feeding studies is like the dietary guidelines diet, or what's recommended that Americans eat just to avoid like chronic conditions. And so those are usually educated or counseled in terms of serving groups, you know, so it encourages like healthy grains and fruits and vegetables and low fat. So that would be kind of the antithesis, I suppose, of a ketogenic diet. And that could be used as a control diet. But I would also, you know, we would want to think about what the outcome is, because we wouldn't want to use a diet that may potentially help another population, potentially Angelman syndrome, who, you know, if they're just having better glycemic control, could benefit. So we just have to consider the population and the question.
And kind of having now been involved both with pharmaceutical trials as well as dietary trials, I think two big differences are that we can monitor if people are cheating on the pharmaceutical trial by measuring drug levels, counting pills, measuring volume of medication left. And how do you really do that? Because in the clinical practice with these diets, we know that sometimes there's cheating that goes on that may or may not be reported. And how do you control for that? I think one of the comments, one of the talks was maybe an ideal population, although be very restricting your patient population, would be study it in kids who are exclusively G-tube fed, because then you're completely controlling what they are getting. So how do you do that? Just kind of good faith that the family and the patient are going to adhere to the diet and while they're in the trial?
Are you specifically talking about feeding studies?
No, even just counseling studies. Because even with feeding studies,
so
I think, you know, you could cheat as well.
Oh, definitely. Yeah, so typically we monitor compliance with food records, but well, actually, usually you would do a surprise 24-hour recall. So, you know, they go about and eat the food that they would normally eat. Let's say it was off study, and then you call them the next day and they report to you what they ate. Maybe it was intentional or unintentional, but that would be, you know, getting a look at what they ate without them knowing that you're doing that. With diet studies, with counseling studies, you're educating them on what to do. And part of them implementing the diet is them keeping track of their diet. So they're already doing food records typically, or at least jotting down how many carbs they're eating throughout the day. And so that can be looked at to assess compliance. And again, there's, you know, the mishap or the accidental slip of them eating too many carbs, or are they completely off the diet? And they know that. We usually have an idea of which one that is, counseling. And then in feeding studies, you usually have like a menu or a record where somebody is marking off everything that they eat or writing a certain percentage of what they eat, and then they submit them. I think in some other trials, we've used a continuous glucose monitor, looking at how blood glucose is controlled, or, you know, changes in the study. And I think that would be really fascinating to have a continuous ketone monitor as well, to see if that plays a role. That might be something interesting in like the whey protein study or the MCT oil study, to see if glucose changes or ketone changes are significant or not. And that can be blinded to the individual.
Awesome. Yeah, the other big difference between pharmaceutical studies and nutrition studies is funding. Because a feeding study, I think, like Gary Taylor, when he gave the keynote address at our dietary meeting several years ago, told me what he thought we should really do is put people in a bubble and absolutely completely control what they eat during the time. But as we all know that have been in this field for a long time, funding in nutrition it's really underfunded compared to a lot of the other funding. So I think that would also, as a community, you know, kind of raise the need that we really do need these trials. So thank you. And then Glenna, I have to say, after watching your videos, I think I could listen to you talk all day. I liked your accent so much. And I thought your talk during this was absolutely phenomenal. I think we work very closely with many of the different patient advocacy groups, and I do think they play such an incredibly important role, not only for people living with this various disorders, but for the community, both medical and scientific, trying to understand it. So what would you suggest kind of moving forward, particularly as a perspective of living with GLUT1 and realizing it's not just the seizures your daughter's dealt with. It's not, and many, the Dravet population, many of these other rare disorders want focus on the whole aspect, not just the symptom of epilepsy. So what would you suggest on how to move this field forward by making future trials more rigorous and unbiased?
Okay. And thank you. I'm honored to be part of this today. And I think with, in terms of rigor, the implementation of the diet in our community lacks a lot of rigor in and of itself. It's implemented a number of different ways, a number of different approaches. We have families in locations where they don't even have dietary support, so they're trying to do it on their own. So for us, a clinical trial would need to be very standardized, very strict guidelines on all the variables that can be manipulated in the diet. The fat sources, the ketone glucose levels, if those are being monitored, timing of the meals, number of calories, all those things, because we really don't have standards in our community about the best approaches. So I think answers for us too on what would be the best approach, the best form of the diet, the best ways to implement it with all the variables that I previously mentioned would be very beneficial, because for our population in particular, we think it's about brain growth and development. That we need to be doing it right, doing it well, and know that we're doing all we can to help optimize that for our patients. When that kind of speaks to trial design, where a placebo for our patients is just, is it even ethical to consider something like that for our patient community? Whether it's a different form of diet. I particularly like the platform approach where maybe, different, I think that would be very beneficial for our community to be able to compare the different types of diet and really get some answers on, what's the best way to do it. And then, just having, if it's something beyond the ketogenic diet, can we ever take a patient off to try a different treatment, to take them off the diet to see? And those are all concerns, that we have in our patient population, especially. And then to speak into the outcomes too, we did an adult patient survey, because like most considered pediatric epilepsy syndromes, although we know it affects patients of all ages and stages in their life, we don't know a lot about the adult experience. So Dr. Mackenzie Cervenka assisted us with a survey last year of our adult patient population, and cognition was flagged as the number one concern and the number one impact on quality of life. And I think that's probably true for lots of the neurodevelopmental epilepsy syndromes, where it makes such a difference in their levels of independence, and just so many things hinge on cognition. And I think for our families, a lot of us have the opinion that we can live with a few seizures if we can come off some medications, and if we can, boost their cognitive performance. Their speech and language is a huge issue in our patient community, and I know in others as well, because it impacts their perceived cognitive abilities. I think sometimes it doesn't always match what they're capable of doing, and that filters through into all kinds of things. The way the opportunities they're given, just the way they're perceived by others, and that limits them a lot of times when they're actually capable of doing much more. So, I think there's a lot of other outcomes that we can look at besides seizures too.
Yeah, well, thank you. No, I think there's a move in the pharmaceutical trials as well to not just look at seizure counts, but look at impact on neurobehavior, neurocognition over time, and especially even in the open label extension trial of those studies, because really it's a short study where you're looking at seizure efficacy, long enough time to assess that. But I think if we can maintain people on these diets, which I think they would maintain if they had good efficacy, then we could judge that with time. So thank you. So Adam's forwarded me lots of very good questions from the audience, and this is a really good question. Is a clinical trial the gold standard to address these issues? Most interventions in medicine are the result of small, well-conceived observations. Clinical trials often negate biologic differences, and it results in varying levels of efficacy, which are rarely followed up on as the outcome sought and found is usually the aggregate result? Is the perceived need for clinical trials a sign of scientific weaknesses, as when the biological mechanism is not well understood? So anyone on the panel want to take on that one?
Well, it was kind of what I was talking about, and I love the question. It was, it was the parachute question, but, you have to be, if you're going to use observational studies, obviously, you have to be incredibly rigorous about other aspects. And I agree that randomized controlled trials, the way I have started to, formulate it in my head, just for people to get a sense, is that a randomized controlled trial says, can you get by bus from Washington to New York? And then it says, yes, you can get by bus from Washington to New York, and here's how often you crash, and here's how long it would take, and here's how many people would get off the bus at the end. But really, what we really want to know is, how should I get from Washington to New York? Should I take a bus? Should I take a train? Should I drive my car? And that answer obviously might differ depending on many things about you personally. Do you have a car? How fast do you need to get there? Do you have claustrophobia, etc.? And we lose all of that nuance when the only question we ask is, can you take a bus from Washington to New York? So, people have to sort of think about that. But, doing anything other than a controlled trial, as Jordan, I'm sure, will tell you, takes an enormous amount of additional thought. And again, I don't think we've given that enough additional thought, the rigor of that.
I love that analogy. It's wicked cool, Jackie. So Jordan, any comments on that?
Yeah, so I think it's an interesting question. When you're designing a clinical trial, obviously you want to enroll the people who are most likely to benefit, because you are really answering the question for that particular population. But it is looking at the effect of that group in aggregate. That's definitely true. But I guess the question of, the question of, is it negating biological differences? It kind of, I feel like, if it doesn't work for most people, maybe it just doesn't work. Are you grasping at straws trying to make something out of this negative trial by looking at, well, this little subpopulation kind of responded, and this, it's usually, perhaps false positives in those areas. I mean, it's certainly hypothesis generating, but I think there's a reason that we do randomized trials, because those are the ones that give us, the most rigorous results.
Thank you. Another question is, one critical thing that seems to be undervalued is taking the person's metabogenomic background into account. This is so important in how a person reacts to specific diets, foods, etc., at the metabolic level. Clinical trials are so expensive, not sure how funding would affect the ability to do this. That's a really good question, and this comes up a lot in many of these trials. What is the difference between the super responders and the non-responders? And how could we tell that with dietary therapy? And I think with dietary therapy, it's probably even more complicated than with a single pharmaceutical agent to really assess that, and what are the variables at play. But anyone on the panel have any comments on that comment?
I can talk. I mean, to be honest, do we know enough as yet about the different aspects to that? And is it a personalized metabolome? And that needs to be addressed in the context of that. I'm going back on the last question. It depends what you mean by a clinical trial. And I'm not being, does it have to be a randomized controlled trial, or do we do a different clinical design? As Jackie has said, we do need to have some degree of rigor in order to address questions. And then the metabolome issue comes in, that we don't know enough yet, I don't think, to actually utilize that. And maybe that does need to be explored. It's following the biomarker question. And if we find that, there is a difference between responders and non-responders, then that is something we can move forward. We've tried to attempt to look at genetic markers and not come up with anything significant. It would be great to be able to predict responders in advance, which we're not able to do as yet. It is still, we have to take our sample and trial and error. So yes, it would be something to pursue, but we need to have some data first before we can explore that on a trial basis.
So just another question for the panel, kind of what trials do we really need? We know the ketogenic diet works. There's 100 years of data on that. Do we need a basket trial to say, is it particularly effective for certain etiologies like Angelman, again, where there's an extreme unmet need for effective therapies? Do we need to compare the current kind of versions of the diet that are in use to say this one is superior to that one? Or do we just need to be prepared as we're moving forward with the idea of whey and all these different things that will likely also need to go into clinical trials? Do we need to have a format for doing those with great rigor? So any thoughts on that?
I have, so some of the questions that I have, all day working with these patients, I deal with compliance and thinking about compliance, and is that why they're not getting effective seizure control? So it's really helpful to that if we have some evidence to show which direction to go after that, like using certain supplements or switching potentially their protein sources. So I think
morning.
that could be helpful.
Okay, well, I think we are running out of time. Do any of the panelists have any final words of wisdom for us who work in this field and trying to figure out the right way to find effective therapies that are safe and well tolerated for our patients, and that will hopefully improve not only their seizure control, but their overall quality of life? One thing I would add, one of the comments I've heard through all the years when people do the diet is families often tell me that their neurologist told them it'd be much too hard for them. And I don't know if that's ever really been assessed, and that could be an outcome variable in a trial, a secondary endpoint on, and did it relieve or increase parental familial stress, to be on this diet, et cetera. So I think if we could really start to think outside the box of what other aspects of dietary therapy can we kind of characterize and quantitate, and helping us decide not only when it could be effective, but in which situations it could be helpful in other manners. So
yeah, I was going to comment on the fact that when somebody wanted to put people in a bubble, that doesn't answer any clinically relevant question. And you just explained why. I mean, you have to figure out whether it works, in the setting with which it's supposed to work.
Right. No, absolutely.
Yeah.
Well, I would like to thank, oh, go ahead.
Oh, I'm sorry. I was just going to say, it seems to me that the diets work, but if people can't sustain them, then that's a major problem. So how to address that is an important thing to figure out.
No, absolutely. And what the variables are. I mean, we do have some children, and Helen does as well, any diet program, they've been on the diet for many, many years because it's been really helpful and the family makes it work. In our experience, it's been harder with adolescents and adults to maintain the compliance. And I think, there's different reasons for that. But I think Mackenzie's experience and the growing experience with the use of these diets in adults, I also think will teach us a lot, and how to make it easier and more effective and better tolerated for people. So I think we've run out of time. I'd really like to thank the panelists. I would really encourage, if people haven't watched the videos, to please go watch them. I think that they were really, really helpful. Made me realize how poorly I really understand statistics. So thank you very much for helping me with that. And again, Glenna, thanks a lot for your beautiful talk on the importance of patient advocacy groups. So Adam, thank you very much. This concludes the first part of the panel discussion.
Elizabeth, we'll be starting up again at 2:15 with our final panel. That's going to be a wrap-up panel chaired by Dr. Susan Messino. So we'll see you at 2:15. Thank you.